Hi everyone, welcome back to the Kitchen Pandemic. My name is Jason Moeller with the Adventure Science Center, and today we're going to look at DNA. Now you will need a few things to actually do this experiment. First, you're going to need strawberries. Because of the cell shape and the DNA structure, strawberries do tend to work best for this experiment, although bananas can also work. You will want a clear plastic bag. You'll want a plastic container. I'm using a test tube here, but glass could also work. You will want a piece of cheesecloth that is large enough to cover the top of the glass or fit inside of a funnel. You will need a funnel. You can either have a funnel at home if you've got one, or you can make one using something like tin foil or paper. And then finally, you will need table salt, kitchen soap, and then rubbing alcohol. The first step that we're going to do is we're going to place the strawberry inside of the bag here, and then we're going to need to smash the strawberry. Now this does take a while, so I've already actually done that for you here. This is what the end result product should look like. The best way to crush the strawberry is to actually just get the strawberries into the bottom corner, take your hand, and crush them this way. Please do not Hulk smash the strawberry against the table. That can break the bag and then strawberries go everywhere. Now, once your strawberry is a nice liquidy pulp like we have here, we're going to add two things into the bag. The first thing that we're going to add is the table salt. You'll want about 1 teaspoon. You can either measure that out, or you can just take this and shake out a bunch of salt into the bag just like that. Once you've done that, close the bag. And then go ahead and use your hand to mix the salt and the strawberry. The reason we add salt is salt breaks down proteins. DNA is a type of protein, so this is going to make the DNA very visible for us when we do the last step. Once you've mixed the salts together with the strawberry, we'll open the bag again. And this time we're going to add some dish soap. 1 squirt of dish soap should do just fine. What dish soap does is it breaks down fats. This is why we use it in our kitchen. The fats here are called lipids, and they help make up the cell walls and cell membranes of the strawberry cells. So in essence, we're, just like we were doing when we were crushing the strawberry, we're using the soap to help break down the cell walls and membranes and make it easier for us to access that DNA. Once you've nicely mixed it, again, you should have this kind of soapy, watery pulp here at the bottom. What we're going to do is we're going to separate the liquid from the remaining pulp that's in the strawberries. To do this, we're going to need a couple of things. First, this is when we're going to go get our plastic test tube, and we're going to get our funnel and place our funnel inside of our test tube. We are then going to go and take our piece of cheesecloth and put the cheesecloth inside of the funnel as close to the bottom as I can. Now, the next step is we're going to carefully open the bag, and we're going to pour the bag of strawberry juice into our funnel and let the cheesecloth do its job, which is separate the liquid from the rest of the pulp. If you're working with 2 people, I would suggest 1 person hold the funnel and have another person do the actual pouring. If you are alone, then what you can do is grab the bottom corner of the bag just like this and tip the juice very carefully. That was probably a little too much on my end. Into the container. As you can see, this can be a little bit messy. Now, this is when we have to be patient and wait because we do need the cheesecloth to do its job. You can speed things up a little bit by taking either a popsicle stick or a pen or even your finger and very gently trying to massage the juice down. Just be careful that you don't work too quickly and push the cheesecloth all the way through the funnel because we don't want any of the pulp getting in. Now, as you can see, the juice is nicely draining to the bottom of my container here. And the best part about this experiment is you don't actually need that much strawberry juice. We have enough already to actually do the experiment. Are you guys ready to see some DNA? The last step of this is to simply go pull this out, grab our alcohol, and then we're going to pour a little bit of alcohol here into the test tube. And what the alcohol will do for us is precipitate the DNA so that we can nice and easily see this. Are you ready? 3, 2, 1. Now, if you look closely towards the bottom, right, and we'll put this right here so it's a little easier to see, you should notice that there is a layer that appears to be a little bit like jelly or gelatin, and I've even had students describe this as snot before. The neat thing about that that's the DNA that you're actually looking at. When you precipitate it out, it kind of does look a little bit like a bubbly snot. And if you have a pen or a wooden craft stick, as we mentioned earlier, popsicle stick, then you can actually go ahead and use that to pull out that nice little bubbly layer that we have right there and look at it even more closely. We do ask, because this has been in alcohol, please do not actually touch it, and definitely do not eat or drink this science. Ladies and gentlemen, I hope you enjoyed that. Thank you very much, and until next time, stay safe.